When people sequence DNA for doing variant analysis, sequencing the entire genome is quite expensive. So a lot of people focus on sequencing only the exons. It's called exome sequencing. You have seen this. When you have done exome sequencing, you need something extra. So you need the genome to map to, of course, but you also need an additional file that contains the location of the exons. And typically what you use for that is a BAT file. It's just a file format that contains the annotation of the exons or the annotation of the genes in the FASTA sequence that you're using for the mapping. Why do you need this? Well, because you've only sequenced the exons, so the reads that you generate, in theory, they should all map to exons or to genes. So you can tell your mapper that. You can tell him, well, I expect that my reads will only map in these locations where the exons are located. Where can you find such a file with exon annotation? Well, typically the provider of the kit that you used for making your exome sequencing library should provide you with such a file. If he hasn't done that yet, you just have to ask.